Hi everyone, we are now on the second lecture for special senses. We're going to be focusing on taste and smell, and then we'll do vision in a separate video. Let's start with a reminder of the functional classifications for those sensory receptors. So we've already talked about the equilibrium and the hearing. Those are mechanoreceptors because they respond to changes in cell shape. So they will depolarize or hyperpolarize depending on which direction the stereocilia are bent. Today we're going to be looking at taste and smell, and we're going to be looking at them together because they both use chemoreceptors, which means they respond to chemicals. And then we'll look at vision, which responds to photons of light. So we'll have the rods and the cones, which are both examples of photoreceptors. Okay, so taste first, a little bit of anatomy review. Here we have the tongue. On the tongue, we have these little mounds that are called papillae. On the sides of the mounds, we have these taste buds. Okay, so this part right here, this is a taste bud. And in the taste bud, it has a little pore where so the chemicals can enter, and it has the taste cells, also known as gustatory cells. Those are going to be the sensory receptors. Now we have the two cell system. So the sensory receptors, the taste cells, will then synapse or connect to a sensory neuron, which will then take it to the brain. Okay, so we have 5 different types of receptors, of taste cell receptors, and each one is responsible for a different taste sensation. We have sweet, salty, bitter, umami. Okay, so for the salty, the chemicals that activate them are generally, they'd make sense, right? If you think of salty, you're tasting salt, and salt is sodium chloride, and so the salt is going to be activated with sodium. Sour, think of the types of things that are sour, and they're sour because they have acids. And what do acids do? They release hydrogen ions. Okay, so sour taste cells, they are going to be activated or depolarized in the presence of the hydrogen ions. Then we have the sweet and sweet ones, things that are sweet. Think of sugars, simple sugars, things like glucose and sucrose, those kinds of things, as well as artificial sweeteners like saccharin. Those are going to activate those cells. Bitter. Now, bitter is a bit more complicated. Bitter is going to activate from a variety of different chemicals. A common one is quinine. And in fact, some people taste quinine and others do not, but that's going to create a sensation of bitter. Now, the one I want to spend just a little bit more time on is umami. So umami is going to activate when amino acids are present. And so what it specifically responds to is glutamate. So the sensation that umami generally elicits is usually referred to as savory. Things that are meaty. And the reason for that is because things that are meaty or savory, they have a lot of proteins. And what are the what are the building blocks, the monomers for proteins? Amino acids, including glutamate. Okay, so glutamate is the one. So you might have also heard of monosodium glutamate. Monosodium glutamate is going to be an artificial man-made way to add the sensation of glutamate into the food. Okay, now we're going to talk about specifically how do they activate. Okay, how do you activate the cells? Okay, so for salty, remember it's going to be activated by sodium. Now sodium is already a positive ion. It's a cation. So in order to depolarize those salty taste cells, all we have to do is allow the sodium to come in through a channel. Here's the ion channel there. When the sodium comes in, because it is a positive ion, it's going to make the internal environment more positive, We call that depolarization. So you'll create a graded potential where you have a positive depolarization. That's going to open voltage-gated calcium channels. And as we've already seen at the synapse of neurons, when the calcium floods in, that's going to trigger exocytosis of neurotransmitter. And then that neurotransmitter will then go and bind to receptors on the sensory neuron, create action potentials, and then we'll take that information to the brain. Okay, so salty neurons, salty taste cells are going to depolarize just because the sodium in the sodium chloride diffuses into the cell. Okay, so for sour activates but with the presence of hydrogen ions. And again, hydrogen ions are positive. So when they diffuse into the cell, you're adding a positive charge. We call that depolarization. That's going to trigger opening of voltage-gated calcium channels. The voltage-gated calcium channels trigger exocytosis of neurotransmitters. And again, that goes to the sensory neurons, activates action potentials that travel to the brain. Okay, so both salty and sour, they're going to activate very similarly. Okay, they're going to be directly depolarized by the thing that they are activated by. Now, bitter, sweet, and umami, they are different. They're a little bit more complicated because they have their receptors on the outside of the cell. Okay, so for example, bitter, one of the things that it responds to is guanine, and guanine is going to bind to a receptor that is located on the outside of the cell. And it's what we call a G protein coupled receptor. You've already seen G protein coupled receptors. We talked about them before, but we'll go through them twice more during this lecture. Once the chemical, in this case guanine, binds to the receptor, that's going to activate a G protein, which will activate an enzyme that causes the creation of a second messenger. Now for this particular one, I'm not going to ask you specifically what that second messenger is because I want to focus on CAMP, cyclic AMP. We'll talk about that later. This is a different second messenger, so I will not ask you to specify what the second messenger is. I just need you to know that bitter, sweet, and umami, those are going to be activated using G protein coupled receptors. Okay, G protein coupled receptors. Notice that second messenger then opens the voltage gated calcium channels, and again we have that release of the neurotransmitter. Okay, so what depolarize the salty taste cells? Salt is sodium chloride. The sodium diffuses into the cell, and that's how we depolarize it. Okay, on to olfaction, which is a fancy way to say smell. So olfaction is again chemoreceptors. And let's do some anatomy first. Here is the nasal cavity. On the roof of the nasal cavity, lining the nasal cavity, we have a specialized epithelium that's called the olfactory epithelium, because embedded in that epithelium, we have our olfactory receptors. Those are bipolar neurons. Okay, so again, one cell system here, not two cell systems. So the bipolar neurons are going to both sense the chemicals and then take that information to the brain. So it'll travel through little holes in the cribriform plate of the ethmoid bone and synapse with the olfactory bulb, which is part of the brain. Okay, so just one cell sensing it and then creating action potentials along the axons that will be conducted all the way to the brain. Now, let's talk about how we activate the olfactory receptors. How do we create that graded potential that will then turn into action potentials on the axon? So they also use G protein coupled receptors, but for this particular one, I want you to know the specific pathways. So we're going to go through it. So here is the receptor. This receptor is going to respond to a particular scent chemical. So this where it says odorant, it's just the chemical that we are sensing, that we're going to smell. So that chemical will bind to the receptor. We call it a G protein coupled receptor because the receptor is coupled or linked to a G protein, so that when the chemical binds to the receptor, the G protein is going to be activated. See how it has a GTP on there? That means it's activated. That'll then in turn activate an enzyme. This enzyme is called adenylate cyclase. Adenylate cyclase, and the reason it's called that is because what it does is it takes monocyclic AMP and it converts it into, wrong words. It takes ATP, adenosine triphosphate, and converts it into cyclic AMP, which is cyclic adenosine monophosphate. I just go by CAMP, right? CAMP. Okay, it makes it campy. So the adenosine triphosphate, the ATP, was converted into cyclic AMP. Now this is your second messenger. Remember that the second messenger, that's the thing that's going to cause the reaction in the cell. The cyclic AMP is the second messenger, and then goes and opens those gated, the cyclic AMP gated cation channels. Things like sodium and calcium will flood in. When you have an influx of positive cations, that's going to create the depolarization. Then from the graded potentials, we will then get an action potential. Okay, so for the olfactory receptors, I want you to know the specific pathway because it's going to come back again when we get to hormones. So you might as well learn it now. So let's look at olfactory adaptation. You've seen this word before, adaptation. Adaptation means that we're going to lose the sensation with continued stimulus. Okay, so basically it means that when you spritz on your perfume and you're walking around, you don't smell your own perfume anymore, even though it's still there. Okay, so we're going to lose the smell with time. So let's revisit why. Why do we adapt to stimuli that are constant? It has to do with prioritizing the information so that more of the brain is devoted to changes that could be potentially dangerous or advantageous, and less of it will be devoted to constant stimuli that is always the same. Okay, so it's about prioritizing change over constant stimuli. So let's review a term here. So if the olfactory receptors are able to adapt, the sensation goes away. Would they be phasic or tonic? Do you remember those words? Phasic or tonic? Phasic, okay, fast adapting, which means they are able to adapt and stop sending signals to the brain. Let's talk about smell amplification. So when we say amplify, it means take something small and make it bigger. And we're not as good as like dogs and things, but we actually are very, very capable at smelling very small amounts of chemicals in the air, right? A tiny amount of chemical mixed into a bunch of air molecules, and we're still able to smell it. The reason for that is because we have signal amplification, because those sensory receptors, right, which would be shown in red here, the sensory receptors are going to activate more than one G protein. Okay, so not just one G protein, but up to 50 different G proteins. And then each of those G proteins will activate their enzyme, adenylate cyclase, which will create even more cyclic AMP, and more cyclic AMP means even more opening of the opening of the cyclic AMP gated cation channels. Okay, so instead of just one single G protein, which is what we see in this picture, we can have up to 50 different G proteins, all activating their own adenylate cyclase. creating even more of the second messenger, the cyclic AMP, and more cyclic AMP is going to open even more of the cation channels. Opening of those cation channels will create a bigger depolarization. More depolarization means we're going to get to our action potential faster, right? We're going to reach threshold faster, and that's how we're able to amplify it. Okay, so that we're very, very sensitive to very small amounts of chemicals mixed into the air. That's amplification. Okay, let's talk about how we can differentiate different smells. Okay, so the reason this is really interesting is because we actually only have about 350 receptor proteins. Okay, and so, but we're able to perceive over 10,000 different scents. So the question is, how can we detect many, many more scents than receptors that we have that respond to those scents? Why is it that we can do, how do we get more than 350 scents that we can perceive when we only have 350 different receptors? And the answer to that is the pattern of activation, because we're going to be able to bind, so molecules are able to bind to more than one receptor, and they're able to bind to them with different affinities. Affinities means attractions. Okay, so for example, let's say we have two different molecules, two different chemicals that we're going to smell. We have molecule A, and molecule A is going to bind to receptor 1 strongly, receptor 2 weakly, receptor 3 weakly. So that pattern of strong, weak, weak, those signals sending to the brain, and it's going to be decoded as, well, that's scent A. Whereas if molecule B has the code weak, strong, weak, that pattern of activation is going to be decoded in the brain as scent number B. Okay, so it's just depending on which of the receptors are activated in order to be decoded as a particular sensation. Okay, that's revision now.